Hello students, this is Dr. Faizan Mirza. I'm discussing with you photosynthetic pigments and the various spectrums that are we having. So when we're discussing photosynthetic pigments, basically there are two types of photosynthetic pigments, chlorophylls and carotenoids. Chlorophylls include chlorophyll A, chlorophyll B, and carotenoids include beta carotene and xanthophyll. Chlorophyll A, if we go for what region of the spectrum are they using up, and how do they appear? So chlorophyll A and chlorophyll B, they appear yellow, green, and blue green respectively, whereas beta carotene and xanthophyll, they appear orange and yellow. Appearance means that this is the light that they are reflecting back. So if beta carotene appears orange, it means that it is reflecting orange color back and it's absorbing all the remaining colors in the visible wavelength. Similarly, if for example chlorophyll A is appearing yellow-green, it means that it is reflecting yellow-green back and it is absorbing every color other than every wavelength other than the yellow and green wavelengths, and so on and so forth. So this brings us to the idea that the absorbance of chlorophylls and carotenoids. So chlorophylls, they absorb in red and blue violet region, whereas the carotenoids they are absorbing in the blue and violet region. Chlorophyll A is always the primary pigment, whereas chlorophyll B, beta carotene, and xanthophyll are all accessory pigments. Now what's a primary pigment, what's an accessory pigment? We'll come to it. We are discussing spectrums here. What are spectrums? There are two types of spectrums that you need to cover: absorption spectrum and the action spectrum. What's an absorption spectrum? This graph here is the absorption spectrum. This graph actually shows you the absorbance of different wavelengths of the visible range, which is being absorbed by different pigments. For example, chlorophyll A, chlorophyll B, carotenoids. You can see in this diagram that chlorophyll A is absorbing in the region between 400 and 500. You see a peak which actually shows you the maximum absorption because of chlorophyll A. Then by 500 it declines and then stays low, and towards the end of the spectrum, beyond around 640, it starts to rise once again, and it goes to a higher value, and then it drops down again around 700. So you can see that which regions of the wavelength are they absorbing. They are appearing yellow and green because yellow and green are the regions of the wavelength that they are reflecting back because they are not absorbing it at all. Similarly, what's the absorption spectrum of chlorophyll B? You can see chlorophyll B is giving you a peak absorbance at 500. So you can just quote this data to tell that yes, chlorophyll B is actually absorbing this much of wavelength, and beyond this, the wavelength is not being absorbed, and towards the end again around 650 it's absorbing this wavelength again. So there are two peaks again. For carotenoids, it's just absorbing in the first half of the visible spectrum. Beyond 550 or 560, it's not absorbing at all. So the two peaks here for the carotenoids comes together in the first half of the visible spectrum. So this is how the absorption spectrum, this is what actually tells you. What about action spectrum? Action spectrum basically tells you the rate of photosynthesis, which is occurring at different wavelengths of light, of the visible light. So how is that possible? What do? We see that in the initial part of the spectrum, this is carrying out a lot of photosynthesis. Then as we go further in the spectrum, beyond 450 till 600, the rate of photosynthesis stays low. After 600, start to rise once again. So there are two peaks. One peak is in the initial part of the spectrum. The other peak comes in the later part of the spectrum. The rate of photosynthesis taken on the y-axis tells you that the initial part of the spectrum between 400 and 450, you have a high rate of photosynthesis, but beyond 650, you have even higher rate of photosynthesis. So if we just go to the absorption, we see that the absorption of the later half of the electromagnetic spectrum was not as high as it was for the first half of the electromagnetic spectrum. So why the first half of the electromagnetic spectrum gave a lower rate of photosynthesis, whereas the later half of the electromagnetic spectrum, having peaks which are lesser than the first half, still were able to give you a higher rate of photosynthesis. So what I'm doing, I'm just comparing this region with this region. Let me just tell you. So I'm just comparing this region. I'm comparing these peaks together, that how these peaks actually are telling you that if the absorbance is high, we should expect the rate of photosynthesis should be equally high. And if the rate of absorbance is a bit lower as compared to the initial part of the spectrum, the rate of photosynthesis according to the absorbance should be low as well. But that's not the case. We see that no, that's not happening. So what's happening here? We see that shorter wavelengths, which are here, they are giving you lesser rate of photosynthesis as compared to longer wavelengths, which are not being absorbed in the same quantity, but yes, they are still giving you a rate of photosynthesis much higher than the initial part of the spectrum. So what's the reason? That's where the physics, where that's exactly where physics comes in. So what's the logic of physics here? What's the logic here? The logic is that we know that shorter the wavelength of any electromagnetic beam or any visible range of light, it would cross the leaf at a very fast speed before the leaf can use it for work done. And why is this so? Because shorter wavelengths have greater energy. So when they have greater energy, a lot of energy is lost as heat, and it's not successfully converted to work done. When it's not successfully converted to work done, so it's lost as heat. Whereas these longer wavelengths, they take a lot of time and they gradually cross the leaf. They are not, the energy is lost as heat. So enough energy is absorbed to be converted into substantial amount of work done. I've already mentioned that reaction center for light-dependent reaction is chlorophyll A, chlorophyll B, beta carotene, and xanthophyll. These are all the accessory pigments. I've already mentioned these points to you. What do they do? What's the function of the accessory pigment? The accessory pigments, they absorb the light and they just harness all the light. They capture all the light that they can and they deliver it to the reaction center, which is chlorophyll A. This is what a photosystem looks like. So what's a photosystem? Photosystem, abbreviated as PS, is actually light harvesting clusters. These light harvesting clusters are present on the thylakoid membrane. You can recall where the thylakoid membrane is present on the chloroplast. So they harness the light energy, and these photosystems harness the light energy and deliver it to the reaction center. So here's this is the reaction center. This reaction center will have chlorophyll A, whereas all these oval black spots that you are seeing here in the photosystem are the accessory pigments, and these accessory pigments are all beta carotene, xanthophyll, and carotenoids. So they absorb a lot of energy from the visible range of sunlight or white light, and they just funnel the light. The light is funneled to the reaction center, so the light energy becomes more concentric. It becomes more energetic. So because it's not lost. Here you are having various photosystems arranged on the thylakoid membrane. The photosystems have two types of chlorophyll A. There is a chlorophyll A in photosystem which will absorb 680 nanometers, so we call that as photosystem 2. And there's a photosystem 1 which is having another form of chlorophyll A which absorbs light of 700 nanometer. So this is how many accessory pigments are there. So when we just compare the accessory pigments, we see that there are several accessory pigments, hundreds of accessory pigments present in a single photosystem. And all these will be chlorophyll B, beta carotene, and xanthophyll, not carotenoid, beta carotene and xanthophyll, and they would just deliver the light to energy to the primary pigment, which will be the reaction center, and that will be only one molecule as compared to hundreds of molecules present here of the accessory pigments. And we know that just beneath this region will be the thylakoid space and thylakoid lumen, and above the photosystem here you will have the stroma. This is how the light energy is actually funneled to the reaction center. This is chromatography. I'll be discussing chromatography in another video, so I'll just skip chromatography for now from here. Structure of chlorophyll. So what exactly is chlorophyll molecule? You don't need to learn this entire structure, but yes, you need to keep in mind that that chlorophyll molecule is having a profiler ring. Inside the profiler ring, there is a magnesium ion, and this magnesium ion is not always in an ionic form. It is having its 2 electrons, and this actually is a profiler ring. And then there's one long hydrophobic tail. And we know phospholipids have 2 tails, but this chlorophyll is having just one hydrophobic tail. The magnesium, we know that it's having 2 electrons, and these 2 electrons play a major role in photosynthesis, in energy transduction. And how so? Because when light energy falls on magnesium, it allows 2 electrons to get ejected out of its outermost shell, and magnesium turns into magnesium plus 2 cation, and these 2 electrons are the ones that will start to move along the electron transport chain, which we'll be discussing shortly. Not in this video, but in the next, or probably in the next after that. So yes, light energy, how light energy is converted to work done in a leaf. For example, if I have a solution of chlorophyll A or chlorophyll B, I have these here, and these are the photopigments. I have just taken out the photopigment. These are not actual chloroplast here. So if this, we know it will be green in color because these are chlorophylls. So if I irradiate it with UV lamp, the UV lamp will allow magnesium to ionize here, so the 2 electrons will be ejected out. When they are these electrons are ejected out, they will start to emit red fluorescence as they fall back into the same electronic shell, and this red fluorescence and heat will be lost from the test tube, showing to you that this was the energy that actually got lost, since this could not effectively be converted to work done. So this exact energy which is being wasted as red fluorescence or heat is the energy which drives photosynthesis. This is the energy which is required for functioning of a photosynthetic system. So when it's present, when these chloroplast molecules actually have chlorophylls, so it's this energy which is trapped and transformed from one form to another, or transduced, it would be more appropriate term here. The energy transduction occurs, and the electrons of magnesium play a very important role, and that's exactly why magnesium is important, for a very important ion for plants, because chlorophylls are made from magnesium. You can just recall what these labels mean. If this is the chloroplast, then you can just try to figure out what this G would be outside the chloroplast. That's it from my side. Thank you so much.